Hello, I'm Dr. Mitton from the Research Institute at Oakland University. I also have a control gene expression lab, and we often will have to sequence DNA products. So if you're a new student in my lab, or if you're somewhere else and you want to use a sequencing service, if you don't have one on your own campus, we use the Iowa State sequencing service. They've been an excellent service providing sequencing for people all over the United States for many years now. On my homepage, kenmitton.com, you can often go to our staff links page if you need a quick link to get to the Iowa State Sequencing Center. Right down here is where you can do your jobs, so you'll have to set up an account with them to do that. Once you have sequencing jobs that you've submitted and have done, you'll have a login and you can go into your account and you can retrieve your sequencing jobs. If you'd like to view sequences, once I'm logged into the facility in this window, you can see that I have some sequencing from some mouse genotyping PCR assays that are complete. And we're just double checking the sequences of these PCRs to make sure they're what we think they are as their target product. You can download your sequencing files and they'll come in two formats. There's a .ab1 file, which is a sequencing data file, the electrophoretic you'll be able to see, and they also give you a .seq file, which is the text of the base calls. Of course, the seq file won't tell you if the base calls are correct or not. You would like to look at the AB1 file to really double check the sequencing that you would trust in your electrophoretograms. You can also download all the files at once with their links down here. And for instance, I've downloaded the AB1 files, and you'll get a folder. It'll uncompress, so that's this folder here. And so here are the AB1 files. Now, if you don't have a DNA viewer or a sequence viewer software on your computer, you can go to nucleobytes.com, and for the Mac they have an excellent piece of freeware that you can install and will let you view AB1 files. So once you install that on your Mac, you'll have no trouble at all viewing your files. And you should be able to just actually click on them. So for instance, this is from a wild type PCR, and it's already launched the 4 peaks, and you can see the sequence viewer. This is the beginning of the DNA sequence. It would have just started right after the primer. It's very noisy, and it might not be able to make accurate base calls, but here you can see the base calls being made by the software. And typically in sequencing, at some point after the primer, the sequencing base calls become very clean. And so in this region, for instance, you can see that there's a clear baseline separation between the G and the C and the next 2 Gs, and the base calls are probably pretty accurate. And sometimes you'll get regions where it gets a little bit iffy, but these electrophoretic peaks are really probability. It's a probability function that it's that base. It's not really looking directly at the fluorescence data. So you can have regions where I'm looking at here, for example, where there's a probability that there's some T-based signal in here, but the probability of it being A's or C's is so much greater that the software is able to call it with some confidence to be what you expect. And so this is just a nice software viewer. You can do adjustments on how wide the sequence is, and you can basically scroll through your entire sequence. And normally you would look through here. This is a PCR product. It's not too long a PCR product, so typically the sequencing will just get right to the last base, hit the end of the DNA, and so this one abruptly ends with an A. So this would be a good sequencing file. There's mostly baseline separation as we go through all the bases here, and you can see this product is really just over 230 base pairs, but this is simply how you can view it.